Okay, next I would like to introduce Dr. Clayton McMahon. He's the Vice President and Director of Research at SarTec.
Good morning, everyone. It's great to see you all here this morning. I've got the pleasure of introducing our second keynote speaker today. And advance to the next one. Our second keynote speaker is Professor Mark Rasmussen, and he works at the USDA. He has a number of different talents listed here on the screen. He has in fact, BS and Masters from Lincoln, Nebraska. He did his PhD work at Illinois Champaign-Urbana, and he also has an MBA from Iowa State. So he's a highly educated man, and we're very fortunate to have him here today to speak about his cutting-edge research that he's doing at Ames, Iowa. His main focus is on microbiology of ruminants, and specifically his title today is going to be Rumen Protozoa, Friend or Foe, and he's starting to peel back the complexity of the interactions between certain types of pathogens that can exist within ruminants and their interactions with protozoa, and so he'll share some of that information today. Welcome, Dr. Rasmussen.
I mostly consider myself a microbiologist, and after last night's experience at the roulette table, at least bugs I can get to reproduce and multiply. Bedding chips seem to just disappear. I can't get them to multiply. I want to thank you for the invitation to come speak, and I hope I've got some information that you'll find interesting and useful. Actually, the protozoa thing I've had an interest in for quite a few years, but I've spent a good part of my time, the last 5 years or so, working on locally what we call the shit detector project. I'm one of the individuals that developed this fluorescent imaging device that the packing plants are now installing for looking at the cleanliness of the beef carcasses as they go through the line. And I spent quite a bit of time on that, but that's pretty much been turned over to private industry now, and we'll see where it goes with their work on it. And so I'm off focusing more back on some of my more original interests, that being the microbes and what goes on inside the rumen of cattle. And throughout our training in this field, we've always thought of Rumen Protozoa as kind of a curious oddity, something to show grade schoolers when they come in. You show them a fistulated cow, and then you take some material and you put it on a slide and put it up on a screen and so forth. But we never really thought too much in terms of this aspect of Rumen Protozoa, that they might actually have present a problem with regards to animal health or animal nutrition. And we're only really beginning to understand that now. I guess before I get into my talk, though, I did want to emphasize one other thing. I'm at the National Animal Disease Center in Ames, and we're undergoing this massive, what I'd like to think, disruption right now, because we're essentially tearing the whole place down over the next decade and rebuilding it. This is a summertime photo of the high containment animal facility that's being built. I've never seen so much rebar in my life in the foundation of this thing, but I guess if you want to build a building that you're going to house things like anthrax and TB and so forth in, you don't want it to crack. And so they've taken some extraordinary construction measures to build this thing. Right now it actually has a big inflated tint over it. Because they'd fallen behind schedule, they wanted to work through the winter. They brought in this huge tent and blew it up here a month or so ago. And so this is all contained now, and this will allow them to continue to pour concrete and work in there this winter. Ultimately, where we're heading with this is with both a high containment animal facility, a low containment, plus a new laboratory. They're going to rename us, rather than focusing on disease, we're going to be a positive spin and be animal health. So this is kind of an artist rendition of what the laboratories are going to look like eventually. although this keeps changing too in terms of the actual design. Just a quick slide to kind of summarize, as I said, some of the things that we've known for a long time with regards to the protozoa and the rumen. They've had a long history with ruminant animals, and there are distinct and diverse species in different species of ruminant. You can go to some of the African ruminants and find different species of protozoa than you do in our domesticated cattle. Their primary purpose, or what they do in the rumen, is they're grazers and predators. They do consume plant material, and people have shown starch granules to be consumed by these organisms, and also chloroplasts from green plant material. They also eat each other. Big ones eat little ones and so forth, and I'll show you some pictures of some protozoa with spines on them, which are thought to be defensive devices. It's hard to swallow somebody if he's bristling like a porcupine. And then, of course, they also consume the smaller critters in the rumen, the bacteria and the fungal matter. And we've always, for the most part, assumed that there's rapid digestion of these engulfed bacteria that they swallow. However, we are beginning to realize now that protozoa can serve as what some scientists who first worked in this field considered them to be Trojan horses of disease. That certain bacteria can get inside of these protozoa, and rather than be killed and digested, actually become stronger and more virulent, and then when they are released, have a detrimental impact upon the health of the animal. This was first demonstrated back in the 70s. Some of you may recall the Legionnaire's outbreak back in the East Coast. And what they found was that Legionella pneumophilia was actually hiding out inside some free-living protozoa that were in the water cooling towers of this big hotel in biofilm. And as a result of that, these bacteria escaped the normal chlorination process that they were treating this water with. And then when they were released from these protozoa, they got blown around in the ventilation system, and a whole bunch of Legion, there was a Legion convention there, American Legion, and got into the air system, and a lot of people became exposed and infected, and there were quite a few deaths. And so subsequent study of this showed this to be the first case of where you actually had bacteria hiding out inside a protozoa. And since that time in the scientific literature, the more people look, the more they find bacteria pathogens following this same kind of lifestyle. And I list here just a short list of bacteria that people have demonstrated the same kind of thing. Francisella, Pseudomonas, Vibrio, Helicobacter, the ulcers organism, Listeria, Mycobacterium, which perhaps is more relevant to the cattle industry. But the more we look, the more we find these kinds of pathogens having the ability to escape digestion and actually survive inside the protozoa. And for the most part, a lot of these bacteria are what we call intracellular pathogens. That is, they thrive on the ability to get into mammalian cells and then wreak havoc. They are able to escape the normal defense processes of that our own tissues put up to kill them and keep them out. I've simply thrown in here some various rumen protozoa pictures. This is kind of our, some people show their kids and things like that. Well, these are mine. Actually, Sharon Franklin, my assistant colleague, is the one who's more responsible for anyone taking these pictures, but I'll claim them as mine too. So on that particular one there, that's a very nice shot of one that's in the process of dividing. That will soon develop cilia down in here as they divide, and you'll end up with two, unlike my poker chips last night. So anyway, as people began to study this, they began to accumulate evidence that it is possible for these to survive and actually grow in some cases inside of these digestive vacuoles in the protozoa. And that bacteria that are capable of doing this come up with protective mechanisms to survive this digestion. And it turns out that those protection mechanisms also tend to be virulence traits, is what we call them. These are traits that allow them to invade and cause disease in animal tissue. Or as one of my colleagues at the lab said, these bacteria, they go into the protozoa like girly men, but they come out like Governator, you know. They become beefed up and stronger as a consequence of this stressful environment inside the protozoa. We've known about benign associations with regards to protozoa, especially in the rumen, with regards to the rumen methanogens for quite some time. And I take this picture because it's the best one that I've seen yet demonstrating this from Carl Davis, who's a professor at University of Illinois in Champaign, where he got a shot of a rumen protozoa just covered with bacteria on the outside. And oftentimes a lot of those are methanogenic. These are the bacteria that produce methane. They closely associate with certain species. And so we've known about this concept for quite some time. But in our research more recently, we began to ask the question, can protozoa be Trojan horse in the rumen? And these little vacuoles here, where they light up, are vacuoles with bacteria. And are they being digested, or are they actually surviving in there and going to come out on another day and cause us trouble? So in our initial research in this past year with regards to this, we focused on an interaction between certain strains of Salmonella and the Rumen Protozoa. And we chose Salmonella enterica phage type DT104. Some of you may remember this from a few years ago. There was an outbreak of salmonellosis up in the New England states in a dairy herd, and the dairy family actually came down with this. And it turned out to be this particular strain, which is particularly nasty because it's resistant to a whole ton of antibiotics. And it turns out that this resistance is on an integron. This is a chunk of DNA in the organism's chromosome that codes for this antibiotic resistance. So what we did was take that strain, and I won't go into too much detail here, just a little outline of kind of what our experiment was. Since we can't grow the rumen protozoa in continuous culture indefinitely, for the most part, we've simply gone out to the rumen on a daily basis or weekly basis and got fresh material. And then we concentrate the protozoa down from a lot of the other feed material and so forth in there. Then we commingle those with the Salmonella bacteria, or what you might think of as the feeding period, when the protozoa has a chance to engulf and take some of these bacteria in. Then after that, we wash away the leftover bacteria. And then we use various means, bead beater and so forth, to bust open the protozoa. And then we recover the bacteria that we get out of the inside. And we take them into what's called an invasion assay. And these are tissue culture of liver cells, which are being used quite widely in microbiology studies because Salmonella is capable of invading these tissue cell lines. And that is our measure of virulence, is how capable a particular bacteria is of invading this cell culture line. And then you have to do some controls. After you allow them to invade, you use an antibiotic with high levels of an antibiotic and kill off all the bacteria that didn't make it in. And then again, you bust them open and you recover those and count and see how many got into this tissue culture line as a measure of just how virulent and tough was that particular bacteria. And so this is some of our data with regards to this. We've got 5 different strains here. This is a chicken strain of Salmonella, which does not invade this particular Hep2 cell culture line. This one, VJ68, is also a non-invasive. EE419 is what's called hypervirulent or hyperinvasive. It's switched on all the time, so all these bars are the same magnitude for our different controls. This is the primary treatment here, where we fed them the protozoa and then brought them back out of the protozoa. This is a control strain which shows some invasion, but protozoa treatment didn't enhance it. So therefore, the conclusion is that although this one survived inside of the bacteria, it didn't become more virulent or more capable. But the most interesting one here is our DT104 strain. And here's the invasion capabilities of in our buffer and so forth, all of our treatments. And here's the one after they went in and came back out of the protozoa. we get about an 8-fold increase in invasion capabilities for that particular strain. And it's right at the same magnitude of this other strain that's switched on in its capabilities all the time. And one other thing I'll note, the cytokinase treatment is actually an active inhibitor, which we paralyze the protozoa essentially, so they're no longer capable of swallowing and taking in bacteria. So that's one of our treatment or control features to show that yes, this is an active uptake process. If we paralyze them, then they can't get in. This is just a fuller span of some of the Salmonella strains we checked against. And what's interesting is all of these, of course, are the ones that show increased capabilities after coming out of the protozoa with the white bar extended out. And it correlates perfectly with whether or not they have this integron. If you'll recall back, this integron is this piece of DNA that codes for this antibiotic resistance on for this bacteria. And so those that don't have that integron we didn't see an enhanced capability. But all of them that did, we see this enhanced virulence capability, enhanced invasion. And so in addition to, it's our preliminary conclusions that in addition to this integron carrying the ability to code for antibiotic resistance, it also has DNA that codes for some of these virulence factors. So there's a lot of things going on in that integron that we didn't understand before. And so just to summarize this particular section, protozoa exposure enhances the invasiveness of some strains of Salmonella. And this invasiveness was linked to the presence of this integron. And the integron is linked to being able to survive inside the protozoa. And here, of course, we've used the cell and tissue culture invasiveness characteristic as a measure of virulence. Well, our next step was to actually, this is the one I wanted to show you, these spines. This is an anti-swallowing device or mechanism for protozoa. Your little roll out of spines that can't swallow you then. The second thing we wanted to do was actually take this out into the animal and see if our ideas held up. And so what we did was we took this DT104 strain that we ran through the protozoa and brought them back out, and we put them into suckling calves. Now DT104 will cause young calves to carry a fever, and the bacteria, they'll go septic too. The bacteria will invade tissues. And so it's a good experiment to try this out and compare our treatment, that is our protozoa treatment, versus those a culture of this strain which hadn't seen protozoa, hadn't been switched on its abilities. And this is basically the summary of our data here. We originally were going to carry this out for 48 hours, but as you may know, all of our experiments have to be done under an animal care and use guideline, and when you reach certain trigger points in your protocol, you have to put the animals down. And so because the temperature got high enough, we terminated this experiment early at 36 hours rather than going to 48. But basically what it showed was that the Salmonella strain that had come through the protozoa gave us higher temperatures in these animals quicker. And also, we recovered at least a log, if not more, bacteria out of both lymph nodes and spleen. These are kind of some of the tissues that the bacteria first gets a chance to invade when it gets into the animal. And we recovered a lot more bacteria out of these tissues than we did with the non-protozoa treated Salmonella. So we look at protozoa now in the free-living world, but also now increasingly in the rumen, as what some people have called as pathogen training camps. That survival of these Salmonella, in particular, within Rumen Protozoa has implications for food safety and also animal disease, and that actually you can increase the virulence of this Salmonella to these calves. And we're carrying on more of these experiments with other strains and so forth to kind of flesh out our results, see if it holds up and other things. So this is, in thinking about protozoa now in kind of a negative sense with regards to the rumen, you can ask particular questions that we need some answers on. And this isn't just for food safety standpoint, which is where our unit at Ames focuses, is on the persistent shedding of foodborne pathogens in the manure, which can end up on the carcass and end up grounding as a hamburger. but also with regards to animal disease. And we're thinking of the mycobacteria in particular, like persistent shedders for yonis, things like that. Do the protozoa in the rumen constitute a reservoir for pathogens? And you've got a scientist also who's interested. One of the interesting things about bacteria is that you stress them, that's when they start swapping DNA, because they're trying to figure out ways to overcome the stress and survive better. And sticking them inside of a protozoa, try and digest them, I guess, is a good definition of stress. And so we're asking the question, and actually has some data on this now, that bacteria can swap DNA for antibiotic resistance if you put two different strains in a vacuole together. So it's got potential in terms of understanding gene transfer under stress. So this gets to the question on a practical basis, what are we going to do about this if this is really the case? And so what I've come up with is this idea of periodic rumen defaunation, kind of like a deworming. You're going to clean these animals out every once in a while. You're not going to keep them clean because they're going to reinfect from other ruminants and across the fence and so forth. But that's okay. Your goal here is simply to clean them out to keep things down. And so will this decrease this reservoir for pathogens and have an effect on incidence and shedding? When do you do this in the production cycle? Do you do it when animals come into the feedlot, when they're weak and more prone to disease, and there's a possibility of them releasing pathogens which can actually then infect and cause disease? Or do you do it from a food safety standpoint at the end of the production cycle before you send them off the market? Or both times? And then, of course, coming up with reliable defaunation methods. The primary methods that we've had for the last 30, 40 years have been research-based. We've had some surfactants and some chemicals that you could use, but never on a large-scale, routine basis. Now, there's some individuals in Australia at their USDA equivalent who are trying to work on a vaccine approach to getting rid of the protozoa. This has primarily been for methane production and increased wool production of sheep. It remains to be seen if this will be successful. Our approach has been to look at antiprotozoal chemicals that are already out there in the drug world and see if they have any potential for use in the rumen. And this is where SarTec comes in, natural plant extracts. And can you use those to accomplish the same thing, to periodically clean the rumen out of its protozoa? And we intend to do a rather systematic search of these agents to see if we can find the best ones and what works and under what conditions do you need to use them and so forth. So we got a long ways to go on this, but at least we're getting started on it. This is one of the larger protozoa in the rumen, larger species. You can see the spine one there. And then finally, I do want to acknowledge the people that have really did more of the work on this while I've been worrying about the carcass imaging detector and construction matters and so forth. Sharon did a lot of the work in my lab, took all these pictures of these protozoa and so forth. Steve Carlson, he's a scientist that works on Salmonella in particular, did a lot of the DT104 work, his technician. And Zoe is a new postdoc in the lab that is, she's been working on the antibiotic resistance and gene transfer mechanisms within the protozoal vacuoles. So with that, I'll stop with my little woolly bear guy here, and if you got any questions, or I can talk to you later today. Thank you.